All right, let's talk about sex. In particular, let's talk about sex chromosomes. Because as we discussed with meiosis last class, sex is pretty awesome. I don't require cameras for this class. Sex is pretty awesome in terms of biology because every gamete you produce is unique. And we're going to talk about not so much sex as a process, we will a little bit, but we're also going to think about those chromosomes that help determine your biological sex. Right? So that's what we're going to be discussing today. What is carried on those chromosomes, how we can use the location of genes on chromosomes to understand recombination, and then we're going to talk a little more in depth about recombination across a couple different examples. So it'll be a interesting class, I guess. I mean, I think all my classes are interesting, but I'm a big nerd, so what do I know? All right, so back to our friend W.S. Sutton. Now, you might remember Sutton from the previous two classes, but Sutton is a really important figure in genetics, and Sutton himself studied meiosis in the great lubber grasshopper. Now, great lubber grasshoppers are kind of interesting. You can Google what they do. I won't talk about it because it's kind of gross, but you can Google what they do. They're a pretty interesting group of grasshoppers. And while Sutton was observing these grasshoppers, he saw the testicles had 24 chromosomes, and then he would see that 22 of those were in matched pairs, so those are the autosomes, and then 2 were unmatched. One was large and one was small, so XY. And that's kind of how we derive our XY chromosome inheritance, for at least for human sex, of course. Now, after meiosis, he found that 2 distinct types of sperm were formed. Half the sperm had 11 chromosomes and an X, and the other half had 11 chromosomes and a Y. And after meiosis, only one type of egg was produced. And so all had 11 chromosomes plus X. So this is the setting that Sutton had, and you know, we're talking about it, so clearly it works, right? And so we can look at the great lubber grasshoppers. This is what they look like, but you can see that fertilization of an egg with the sperm carrying the X gives you an XX female. Fertilization of a chromosome of an egg with the sperm carrying a Y gives you an XY male, and thus Sutton concluded that the X and Y chromosomes determine sex, and clearly that's true, right? We know that XY chromosomes are the sort of the ultimate determinant. They're at least the presence of the Y chromosome is the ultimate determinant of whether you're not going to develop into a biological male or a biological female. And not just for grasshoppers, but obviously you guys all know, and you saw in your little VGL lab last week, that XY chromosomes also determine sex in humans. So, and that's kind of the way it goes, right? You should have a general idea that your mother always gives you the X chromosome, right? She has no inability. She has the inability to give you anything else but an X chromosome, but it's really the sperm that determines the sex. And so, globally speaking, when we look at this sort of system, it's about a 1-to-1 ratio of male to females. And if you look at the global population of humans, it's slightly skewed towards males. It's like 49.9% females and 50.1% males. So it's pretty close to a 1-to-1 ratio. There's some interesting hypotheses as to why there are more males than females. From a biological context, of course. I mean, there's all sorts of issues socially where people drown little girls because they want boys to carry on their name. But biologically speaking, we do know that Y carrying sperm are slightly faster than X carrying sperm. So there's hypothesis out there that males are slightly more abundant in the population because the Y chromosome makes Y carrying sperm slightly faster. Thus, they have a higher probability, not much, but a slightly higher probability to fertilize the eggs. So I think that's kind of cool. But again, there are also other, social issues that are surrounded why there are more males than females. So now, the reality is it's not so much the Y chromosome itself. It's actually just a really small portion. And it's called the SRY region, or the sex determining region. And it's really, really straightforward. Now, we can look at XX normal, right, and XX females, XY males, and their SRY plus for males, and that produces a normal biological male. But you can have sex reversal, where you can have XX females, where they're SRY plus, or XY female. I'm sorry, XX males, which are SRY plus, XY females, which are SRY negative, or XY females that are also SRY negative. So it depends, but you can doesn't necessarily mean you have to have XX to be female, right? Or XY to be male. Do you have the SRY region on your Y chromosome or potentially on your X chromosome? And that's really what determines the difference. And I think that's kind of a really interesting concept, that it's not so much the presence of chromosomes, it's the presence of a specific gene. Now, you could think of biological sex. We're speaking in this class about biological sex first. We're not talking about gender. We're speaking about biological sex. And you can think of biological sex as almost like a trait, right? Male, female, right? Testicles, vagina, right? That sort of dichotomy. And at the end of the day, it's this clear genomic region, could be carried on the X or the Y, and determines this really complex trait, right? And it's not just, testicles, ovaries, right? It's, all the things that come along with being a female, reproductively, and the same thing for males, and then biologically, physicality-wise of being a male versus a female, right? So it's a really interesting genotype-phenotype connection, where simply the presence of this determines whether or not you essentially have the traits of a male and female. Yep. What's your question, Abigail?
So, I know sometimes babies are born with undeveloped parts. Would this be like a case of that, or?
No. So undeveloped parts are controlled. Well, it depends on what it is, obviously, but by and large, it's a developmental issue. So kind of like meiosis and mitosis, where a predictable series of events have to happen. Developmental disorders like that are simply something along the way didn't get checked. So a box didn't get checked, and testicles didn't get developed, or there's these weird conditions where the urethra will come out at the base of the penis as opposed to at the tip as it's supposed to. So there's all reasons for that. They're partially genetic, but mostly developmental thing. Something was turned on at the wrong time, or something was turned on slightly too soon, or not at all, or too late. So development in humans is actually development in anything for that matter, is a really complex process. And there's lots of things that had to go right for you to look like a normal human being. So, but I think that's a good question. Now, again, SRY is really determining whether or not you develop testicles or ovaries. And the reality is, homology speaking, testicles and ovaries aren't all that different. They're gonads. And so it's just simply the processes that come along with developing testicles versus ovaries. And once you develop one or the other, you kind of have this, change in chemistry in your body, which dictates, you becoming a male. You can just look at how similar the body plan of males and females are, right? It's really similar except for the nether region, right? Right. So, but yeah, so that was a really good question. It's developmental issues are tough, and it's mostly a transcriptional thing, but it can be a genetic thing. So something transcriptionally is not being turned on properly. So now, the interesting thing about the Y chromosome is it's kind of, a lame duck chromosome. It doesn't have a whole lot in terms of material. Now, the X chromosome, as you can see, has 850 genes, and the Y chromosome has about 50 genes. Now, that's not a lot, right? 50 genes is nothing, right? Think about the human genome. It has 22,000 plus genes. So you have this really tiny chromosome, but it does play an important role in the development of biological sex. Now, at the end of the X and Y chromosomes, we have what are called pseudo-autosomal regions. And as you can see, autosomal being like an autosome, and pseudo meaning like they mimic an autosome. And the reality is these PAR regions, or pseudo-autosomal regions, simply allow these to pair during meiosis. Now, if you cut off PAR1 and PAR2 on one of these chromosomes, they actually couldn't pair during meiosis. Thus, it just wouldn't, meiosis would fail, right? You would have a cell with no sex chromosomes, right? And that's clearly going to be deadly to this individual. So that's kind of an interesting region. Now, the females have lots of lots of good stuff on here. 850 genes is no small number of genes that in terms of size. Now, the X chromosome is a bit smaller than your average chromosome, but it's still pretty sizable. Now, the our friend here, the Y chromosome, has what we call the MSY region, which includes a couple different things. Now, it includes SRY, which again determines biological sex, and 3 male fertility genes, and as well as 8 essential genes shared with the X chromosome. Now, the reality is 8 of 850. So if you have a recessive gene here, there's likely no corresponding allele on this chromosome outside of the 8 that are present. So the reality is the Y chromosome basically has nothing on it. That's the way I sort of think. I kind of think it was like a blank. While it's great for developing a biological male, it doesn't have all that important genetic information that the X chromosome has. So now, there are other types of ways to determine biological sex in the animal kingdom, and you might have ran into one of these when playing with the virtual genetics lab, and we'll get to it in just a second. But in Drosophila melanogaster, so an organism we're going to be using in lab this year, the ratio of X chromosomes to autosomes actually determines gender. So if it's skewed one way or the other, it becomes male. Skewed the other way, it becomes a female. Now, in humans, the presence not so much of the Y chromosome, but of that SRY gene determines, biological sex. And it's kind of interesting. Abnormal numbers of X and Y chromosomes have different effects in humans and flies. Now, as we sort of mentioned, generally speaking, if you have too few or too little chromosomes, you kind of are in a tough spot as a cell. Most times you have a trisomy or you're missing a chromosome, the cell typically dies. However, there's a little bit more wiggle room in terms of sex chromosomes, which I think produces some really interesting phenotypes here. Now, we have this nice contrast between Drosophila and humans, and Drosophila being one of the most important model organisms to understanding human biology. Now, XXX individuals produces, non-viable Drosophila, but it produces a nearly normal female. XX females is normal in both. XXY Drosophila is a normal female, but in humans it produces what we call a Klinefelter male. They're sterile, they're tall and thin. So not the best of disorders, but certainly not the worst. XO, i.e. they're missing a Y chromosome or a second X chromosome, produces sterile males in Drosophila, but in humans it produces Turner females, which are also sterile and have a short and webbed neck. I was trying to think of good ways to describe what a webbed neck looks like, but definitely Google what Turner syndrome looks like. It's the phenotype is weird for the neck part at least. XY is normal males, XYY in Drosophila produces a normal male. In humans, it's a nearly normal male. Almost. A little bit weird, but not too crazy. And if you have no X chromosome, but you have a Y chromosome, you're toast in either species. So kind of this interesting, flex, right? You can have one extra chromosome or one too few. And what really seems to be the determinant is, do you have that X chromosome, right? How many X chromosomes you have? Because clearly without an X chromosome, you're toast. Extra Y chromosomes, not too bad, right? So XXY males, not the worst genetic disorder. So there's kind of this wiggle room in terms of what your phenotype will be just by the presence or absence of one or more X or Y chromosomes. Now, in terms of sex determination, we have a couple different terms. So we have heterogametic sex, which is sex with two different kinds of gametes. So XY for humans and ZW for birds. Hemiogametic sex, where sex with one type of gamete, i.e. XX females or ZZ males in birds. And in some species, the environment actually determines your biological sex. So lizards, alligators, tortoises, turtles, and some fish, well, our reptiles, it's all determined by temperature. So cool temperatures produces females, warm temperatures produces males, and vice versa in tortoises. Fish, older adults produce females, younger adults produce males. That's kind of an interesting system. Bees and wasps, it's either haploid or diploid. So diploid is female, haploid is males. And then you have this, the standard XXXY system. But you have the ZZ-ZW system in birds and butterflies and many other species of insects as well. Whereas in humans, right, XX is, female. In this case, the corresponding genotype ZZ produces males, and ZW, which would be corresponding to XY, produces females. So it's kind of the flip of what we see. So instead of males having one useless chromosome, in a ZZ-ZW system, females have one useless chromosome. So you might have run into this one at some point in VGL. It's not, it's just a flipped version of our system. So nothing crazy. Now, couple terms, couple definitions. We have our germline, which are specialized diploid cells set aside during embryogenesis. So cells that are then going to go on and produce more sex cells. And then we have gametogenesis, which is gamete formation, right? And that's that's it's at its backbone is meiosis, but it also has some specialized events before and after meiosis. And so we're going to talk about this for a little bit because it's an important part of genetics. But different types of animals have variability on this process in terms of gametogenesis, and we're actually going to talk mostly about humans because you guys are mostly interested in human things. Now, in humans, we have oogenesis, which is one of my favorite words to say. It's just I like anything that has a double O like that in the front. Produces one or more ovum from each primary oocyte, and human spermatogenesis produces 4 sperm from each primary spermatocyte. And you've probably seen some sort of idea about this before when you looked at maybe in biology 1, back in the day when you were young, youngin. Now, oogenesis, again, the production of the female sexual gamete. Now, oogonia, which are diploid germ cells in the ovaries of female embryos. Now, these oogenia, or genia, or however it is called, I never know. They divide by mitosis and become what we call primary oocytes. And then they arrest in what we call diplotenia of prophase 1 before they physically enter meiosis 1. So basically, they divide by meiosis. So we start off with oogonia. They divide by meiosis to become those primary oocytes. Then they halt. They basically just hang around for an extended period of time. And then they enter meiosis 1 when the time is correct. Now, meiosis 1 then produces two cells, one large secondary oocyte, and then a smaller sister cell, which we call the first polar body. And polar body will come up in a second. Now, at puberty, one primary oocyte per month resumes meiosis at ovulation, and this is what we obviously call the period, that time of the month, on flow, whatever you want to call it, for ever want to call a woman's reproductive series of events once a month. You call whatever you want. Now, during this, we have the completion of meiosis 1, producing a large secondary oocyte, and then the first polar body, and then a second oocyte arrests in metaphase 1 of meiosis 2. Now, after fertilization, meiosis 2 is then completed and produces a mature ovum and a second polar body. So, kind of a weird system, but let's look at this just conceptually speaking. Now we have mitosis, so this is what we're seeing occurring in the fetal ovary. Now oogenia, remember, produces 2 primary oocytes here, and then they go on to division, producing arrested primary oocytes. Now, meiosis 1 occurs, so remember this starts at puberty, and meiosis 1 occurs, but it does asymmetrical cellular division. So remember, it produces this first polar body, and then it produces a much larger overall cell, and this is what we call the secondary oocyte. And then meiosis 2 eventually does not allow this smaller oocyte to essentially divide, but this larger one divides, producing just a second polar body as well as the mature ovum. And speaking in terms of biologically, or I guess I could say physiologically, the primary oocyte is within the follicle. It develops, then the mature follicle within the secondary oocyte, and then you have the rupture follicle, that thus releasing the secondary oocyte, thus having the capacity for that to be fertilized, right? So that's what we're going for. Not the, kind of an interesting look at what happens. Spermatogenia or jonia, this is where we produce sperm, so it's found only in testicles, and similar to the production of eggs, we have diploid germ cells divide by mitosis to form primary spermatocytes. After puberty, the maturation then begins to form sperm. Now, unlike females, there's no really, extended arrest at a period. Biological males are constantly producing sperm from the day they hit puberty till the day they die. That's just kind of how they go. Now, unlike our eggs, primary spermatocytes undergo symmetrical division at meiosis 1 to produce 2 haploid secondary spermatocytes, and then those secondary spermatocytes undergo another round of symmetrical division at meiosis 2 to produce 2 spermatids. Those spermatids then mature to become sperm, and as we discussed earlier, there's about a 50-50 split between X sperm and Y sperm. Or if you're one of those kind of odd males where your XX should be 100% X, right? Just depends. And this is the this is the process we're going through. Mitosis, so this is the spermatogonia. Then we produce the primary spermatocytes, which then produce secondary spermatocytes, and then produce spermatids, and then they mature into the sperm that we've all seen before. Now, in terms of what has to happen, they get slightly elongated, and as you can see, they have a flagella. So they have not a ton of crazy things that happen to them, but there are some key morphological differences between spermatids and sperm. And the entire process of spermatogenesis takes males approximately 48 to 60 days, so not a lot of time, especially considering humans live for decades, so it's not a lot of time. But billions of sperm are produced over a lifetime. So as I mentioned, if you're a male, you produce sperm starting day 1 of puberty, and you don't stop producing sperm until after you're dead. So just the way it is. Now, that's how we make the birds and the bees, I guess. Now, we're going to talk about a related process in terms of our sexual chromosomes, which we call sex linkage. And this involves those genes that are located on those XY chromosomes, and it's mostly, obviously, the X chromosome. Now, our early 20th century geneticists, so Hutton was a part of this group. I'm sorry, Sutton was a part of this group. They tested for the transmission of characteristics other than sex via the transmission of sexual chromosomes. But we really didn't get anywhere until T.H. Morgan actually started using Drosophila melanogaster, which we're going to use in lab, and it's also, a model organism for understanding many processes. But he used Drosophila because it had really short generation time and had produced large numbers of individuals. And so they used Drosophila to track sexual chromosomes, to track how things were passed on from one generation to the next, to understand this idea of sexual linkage. Now, we can look at some nomenclature for Drosophila genetics, and it's not all that different from what what you've seen thus far, but we'll just go through it one more time. Now, the gene symbol name for any mutant phenotype that is associated with a gene. So in this case, we'll talk about white normal gene, the normal white gene producing white eyes. Now, the wild type allele is always denoted with a superscript plus, and the recessive mutant is denoted with lowercase. So, for example, in this case, white eyes, the gene for white eyes is just W. The wild type is W plus, and the recessive would just be W, little w. Dominant mutant alleles are denoted with uppercase. So, for example, the gene symbol for bar eyes is just BAR. The wild type allele is still BAR plus. Go away, go away. And the mutant allele BAR simply has the mutant abnormal eyes. So very sort of not all that different, right? The wild type has always got the plus, and if it's recessive, it's lowercase. If it's dominant, it's uppercase. So nothing new. But we can look at some data from Morgan. So this was in 1910, so 110 years ago at this point, which is kind of weird to think about, but it's a long time ago. Now, he discovered what we call white-eye Drosophila, and you might have seen these before. They're white eyes relative to the wild-type red eyes, so nothing crazy. Now, He did a couple things. Now, he crossed a white red-eye female with a white-eye male, and the progeny was all red-eyed. And then he tried another cross where he took red-eyed females and red-eyed males. But you'll notice the difference here is we have wild type here, recessive here, and over here we had double wild type. So, it's you can think of it as big A, little a, right? If you wanted to, but these are denoted with X's because they're on the X chromosome, right? So it's an important thing for denoting this. But you'll notice that in Morgan's case, he had this heterozygote. When he crossed it with a male, he had 3 red eyes, but he had that 1 white eye. And you'll notice that that white eye was male. And that's kind of a standard thing we see in sex linkaged genes that are linked on sex chromosomes, where males are most common, not always, but most commonly display the trait of interest or the mutant trait of interest. Because remember, that Y chromosome only has about 8 genes on it that match with the X chromosome. So unless it's one of those 8 genes, that Y chromosome basically serves as a nothing. Now, we can also look at what we call crisscross inheritance, and this also occurs with our sex-linked traits. So you'll notice we have our XY male, that's white eye, and we have basically a heterozygote, right, plus little w. So it's basically a heterozygote. And genotypically speaking, we have some unique genotypes here, but you'll notice we have 50-50. Now, we can actually look at, again, this is what we call crisscross inheritance, where we have a swapping of genetic traits between sexes here, where we have a white-eyed female crossed with a red-eyed male, and you'll notice that all the females are red and all the males are white because it's crisscross. And why is that? Remember, males always get their Y chromosome from their dad. So if a dad donates this, the mom can only donate this. Now, conversely, females always get 1 chromosome from dad, 1 X chromosome from dad, and the other one from mom. So in this case, the dad is always donating a W plus, the mom donating a little w, thus they have to display the dominant phenotype. So this is what we call crisscross inheritance. It's really nice to see this pattern actually work out, not just in a scenario like this, but also in a laboratory setting. It's kind of a fun experiment to do. So now, from this idea that we've seen cross A, B, C, and D, Morgan concluded, and as I've already told you, that the white gene had to be X-linked, so it had to be carried by this X chromosome. Now, because males only have 1 of those X chromosomes, they have half the number of alleles as females with 2 X chromosomes do. So this gives us a new term, and this may be new to you. I'm not sure. We have hemizygosity, which simply refers to the reduced allele number during sex linkage and the possession of a single sexual chromosome, X or Y, rather than a heterozygosity of autosomal alleles. So that's hemizygosity. Now, we can actually use what we call rare mistakes in meiosis to help actually confirm what we see from Morgan. Now, Charles Bridges found that about 1 in every 2,000 male progeny of white females have red eyes. And so what does that look like? Well, we have white eye females crossed with red eye males. Now, normally speaking, you had, we have meiosis occur where we produce the red X here and the absent Y chromosome here for the sperm. But you'll notice that the females was producing sometimes about 1 in every 2,000, 1 embryo that was XX and 1 embryo that was completely, nothing. It had no sexual chromosomes, right? So we had this non-disjunction error. And when you do a Punnett square of that, you get XXX, but that dies, OY, which also dies. But then you get males that are XW plus, red eye, and then XXY females that had white eyes. So you had this kind of flopping, right? Why? Because we had non-disjunction occurring. And again, this phenotypic ratio only exists in this very special 1 in 2,000 chance. Now, Bridges hypothesized that these red eye males arose from mistakes in chromosome segregation during meiosis in white eye females, and he termed these non-disjunction. So we already talked about non-disjunction last class, but The term dates back to observations seen in Drosophila embryos. So a pretty cool little a little tidbit of history in terms of how non-disjunction was coined, how it was first observed. But again, an important way to essentially confirm chromosomes, right? When we're thinking about this, we didn't quite have all the evidence. Adding more on top of it, we can start to confirm chromosomal theory. We can also see a similar process here where non-disjunction was producing XXY females that could have their chromosomes traced. So, the males are, doing their normal thing, but we can have this sort of weird sort of case where we'd have an XXY female. It would produce XY, XXY, and then you have this sort of this odd-shaped Punnett square. But you also notice that there's going to be, as has been denoted on this figure, that XY and XW are more frequent than XX, XXW, or just Y. But again, you can see we have these distinct phenotypes, and then we have, 2 out of 8 essentially kicks the bucket and dies. So what this tells us is that they segregate distinctly in 2 ways, producing progeny with unusual sex chromosome complements, thus providing more evidence that sex linkage and the chromosome theory is a thing. Now, again, this is back in the early 1900s, right? This is just showing you why sex linkage sort of works, how the chromosome theory works, and all that fun stuff. So that's kind of where we are in terms of sex chromosome. Now, when we successfully predict the movement of chromosomes based on Drosophila phenotypes, this provided evidence that specific genes existed and were transmitted via specific chromosomes. And again, it seems like kind of a meh thing now, right? We know that, right? We know chromosomes carry genes, but again, this is early 1900s. We didn't know that. Now, this all has been, nicely knitted, what we've discussed and what we discussed in the previous class and the class before that. It's all been nicely knitted under the chromosome theory. And this also explains how mutations can be heritable, even if they occur only in germ cell lines. I'm sorry, only if they occur in germ cell lines. Right? You can't have a mutation occur in your throat and then be passed on, right? It has to occur in your primary. It has to occur in your germ line to then be, passed down to in the lines of germ cells. So let me give you an example of this in humans. So the best study, or the easiest to understand example, is red-green color blindness. And it's inherited as an X-linked recessive trait. And so normal people see the Earth like this, but those that are have red-green color blindness actually interpret the Earth like this. So it's kind of an interesting dichotomy. They lack the capacity to see that green. So it kind of looks like the planet's dead on this side. I think this is a really great representation of what red-green color blindness looks like. But that's an example. And there are many other examples of sex-linked traits, hemophilia, and so on and so forth. So it really depends. Now, we can actually use our pedigrees that we discussed last class and the class before to understand how things are inherited based upon sex linkage. Now, we can use X-linked recessive, and we basically see that when something is X-linked recessive, the mutation never passes from father to son, and then the daughters of affected males are carriers. And half of the sons of a carrier will inherit the trait. So again, if we're thinking about color blindness, we have a colorblind father. Half of the sons of the father carriers will inherit this trait. Now, if we have X-linked dominant, the trait is seen in every generation, and then the affected males will produce affected daughters and unaffected sons, because remember, they get their Y chromosome for dad, they don't get any other genetic information but that Y chromosome. And if there's Y linkage, which is particularly rare, there's only 50 genes on there, so it's a particularly rare thing, but it's something that's only obviously could be seen in males. I had an example of Y-linked traits in my head, and I've gone and forgotten it. But you can take a peek on the internet to see what Y-linked traits are. Nothing too common, as you can imagine. But let's look at a color blindness pedigree. So we have the dominant gene for the female, so XC, XE, and then we have the males XC, Y, and you can see the male is a presenter of the trait, their offspring, which they produce two, one male and one female. You notice the male is completely normal, and the female is a carrier of that gene, right? Because she has to get this X chromosome from dad, this X chromosome from mom, and you can see the same thing for the males. Has to get the X chromosome from mom, Y chromosome from dad. Now, when this carrier has offspring, you'll notice that the male, again, has no gene for color blindness. Thus, it passes its color blindness. It passes its Y chromosome to its son, and the son has a 50-50 chance of getting either one of these chromosomes from their mom. In this case, he happened to get the recessive allele from mom. Thus, he got the trait, and then his brother, again, got Y chromosome from dad, and he happened to get the other non-recessive chromosome from mom. Thus, he is not colorblind. So we can use our pedigrees to really see that, but let's look at a much sort of expanded pedigree. Now we can actually look at a real life example. So this is hemophilia. So hemophilia is recessive sex-linked trait. It's called the royal disease because of this pedigree that we'll talk about in a second. But those that have hemophilia, as many of you probably know, you simply cannot clot properly. So even if you get a tiny cut, if you don't get proper treatment, you would die, right? You would just bleed out because you can't clot your blood. Thus, a paper cut would mean you're doomed. Now, modern times, you take clotting factors and you're A-okay. Back in the day, if you had hemophilia and you got cut or you got bruised, you would be toast. Now, those that had hemophilia in the normal population were easily weeded out because, if you're a farmer, you're going to get bruised, right? If you get bruised, you potentially die, right? So average everyday people, not very common hemophiliacs at adult age. But when you start to look at Victoria, England, or any royal line, they don't do too much. They kind of just sit on their butts all day. And that's not me just being snooty. That's just kind of the reality of this time period, right? If you were royalty, you didn't do a lot of stuff for yourself. Hell, you probably didn't even get dressed yourself. So the probability of having hemophilia and not dying, it was much higher if you were royalty. Now, we can look at this. We start with Queen Victoria. She married Prince Albert. She was a carrier, and Prince Albert was completely normal. And then you can see their entire set of children, from Victoria to Helena here. There was one carrier. We had Beatrice here. So, none of their kids were affected. Now, you'll notice that we also had one of their children, Leopold, was a hemophiliac. And he married Helena, and they had normal children, except for one child, which was a carrier. And she married someone, I should say, and they had one child that was hemophiliac and two that were normal, non-carriers. And we can see the same thing over here. Alice married Louis IV, and then one carrier, one displaying the trait, and so on and so forth. So again, X-linked recessive. And this is what you see simply when something is recessive and X-linked. You'll notice that all the people that have hemophilia are males. All the people that are carriers of hemophilia are females. Remember, in a sex-linked trait, males simply cannot be carriers. So we can look at just an example of an X-linked dominant trait, hyphosophatemia. I believe I said that right. You'll notice that there's some parental generation. All the children have it. Thus, one of the parents had to be a carrier, or I'm sorry, expressing this trait. And you'll notice how much more prevalent the trait is throughout this entire pedigree, right? It's complete exact opposite of what we hear, right? Where we see very few that have the trait. In this case, we see basically a 50-50 split, or about half of them have the trait and the other half don't. So very different, very, very different inheritance patterns between these two distinct dominant versus recessive. And this idea, right? This is not just unique to X-linked traits, right? This is the same pattern we would see inside of any recessive trait or any dominant trait, right? So these patterns aren't unique, right? Now, there's a really interesting phenomenon that occurs, which we call dosage compensation. Now, human females have two X chromosomes and male only have one, and that's what we've been talking about for the past, 30 minutes. Now, dosage compensation, it's interesting, but what it does is it simply offsets what would be double the gene products in the homogametic versus the heterogametic sex. So remember, homogametic is XX, heterogametic is XY. Now, in female cells, to compensate for having extra genetic material on that other X chromosome, they actually inactivate one of those chromosomes. And they do it with a special type of RNA to do it. You'll have to take cell biology to learn that if you haven't taken cell biology already. But the inactivated X chromosome is condensed into what we call a bar body. And you can see the bar body here. It's just this little dinky piece here. And again, it's inactivated. So whereas in males, the X chromosome is being expressed. Remember, there's no corresponding genes on the Y chromosome. In this case, the X chromosome, one of them gets completely deactivated and the other does its thing. Because remember, when we think about genetics, right, we have two alleles on both chromosomes. Only one is expressed at a time. And so, but the X chromosomes have to actually be physically turned off because they're always going to be wanting to express. So this is what we call dosage compensation. Now, human females also can do some really cool stuff, which I think is one of the more remarkable things about human biology. But let's take a peek at it. So they can actually have what we call a patchwork, and it all arrives from this idea that during early female embryos, each independently activates one X chromosome. So sometimes, if you're producing cells, you might inactivate this chromosome versus this chromosome, or so on and so forth, right? So that's what we're going for here, right? Different parts of your skin, different parts of your body as they develop decide to inactivate one X chromosome over the other. So what you can have is actually these patchwork females, where you have some parts of your body are expressing one allele and other parts of your body are expressing a completely different allele, which is kind of a wild thing to think about, right? That you could have this really different sort of phenotype across your body. And you could also, I can't remember the example I had off the top of my head. I'm bad at examples this morning apparently, but the idea you can have like potentially different skin pigmentations or different physiological traits at different points of your body, which is kind of a wild thing to think about, right? That maybe your top of your scalp behaves slightly differently than the bottom of your feet. So I think that's kind of a cool aspect of being a human female. Now, we can look at what we call sex-limited traits, and these affect a structure or process found only in one sex. Now, in Drosophila, there's a mutant called stuck mutant males, and they actually can't separate from females after mating, so their genitalia just kind of prevent them from getting out. So these only affect males. However, there are what we call sex-influence traits, where they appear in both sexes, but hormonal differences may cause a difference. So, for example, male pattern baldness is a great example of this as well. So we have John Adams, one of our founding fathers. He had a very different type of baldness, and then his son John Quincy Adams had a very different type of baldness. Ugly men, but obviously our second president, one of the founding fathers, clearly important men, but man, they was ugly. But again, male pattern baldness is pretty common. So like, for instance, I'm losing my hair and probably a way similar to John Adams here. My sister, same genetic line, has no evidence of hair loss. So it influences different sexes differently. So, and that's what we mean by sex influence traits. Now, our sexual organs develop from 4 groups of precursor cells inside your embryo. Now, 2 of these groups are what we call bipotential cells, and they can develop as male if the SRY gene is present, or the SRY region is present, or female if the SRY is absent. And so these are basically the precursors to gonads, and they produce the external sex organ precursors as well. So again, we start off all the same. It's the flick of the switch of the SRY region. Is it present? Is it not? That determines whether those precursors develop into testicles, and a penis, or a vagina and ovaries. Just depends. And all the other stuff that comes along with being a female, of course. And the two groups themselves are specific to each sex. So female internal sex organ precursors degenerate in the presence of SRY. Otherwise, they will develop into female sexual organs. Male internal sexual organs precursors are the opposite. So simply just reverse that exact statement I just said. So again, very similar developmental trajectory, flick of the switch, which is why you can have an XX male or an XY female, because again, it's all about that flick of the switch of that SRY region. And this is just kind of a handy dandy table with a penis on it that you can look at, what the sort of development is if you're interested. But I'm gonna keep it on because it's not the most important thing. Now, you can also have what we call intersexuality that arise from mutations in genes that are downstream of SRY. Now, there are genes that promote or inhibit sexual organ development, and they can produce phenotypes in XY individuals that are a combination of male and female characteristics. If you have your textbook, it's figure 415 from your textbook. Just as a note, I don't know if anyone bought the textbook or is using the textbook, but it is figure 415 if you are. Now, we can have what are called AMF hormones. These inhibit female sexual organ development, and the inheritance of a mutant AMF receptor results in complete male sexual organs plus internal female sexual organs, which is kind of an odd thing to think about, but that happens. Now, if we have the inheritance of a mutant androgen receptor, which is an X-linked gene for testosterone, this results in testicles that the default internal female sexual organs in neither male nor female internal sexual organs. So it's kind of this weird thing. So let me break that down. It's kind of a weird thing. So they have just testicles, female sexual organs, but no internal sexual organs. Kind of a weird thing to think about. But it is what it is. But at the end of the day, as I mentioned sort of at the beginning of this class, sex, biologically speaking, not gender, but sex is morphologically a trait. Morphologically, it's a trait. Now, this idea of intersexuality is demonstrative of sex being non-binary traits like many traits, right? So this sort of variability of how your genes control your development of testicles and a vagina, right? It kind of shows that it's this non-binary trait, right? It's not male versus female. It's like there's this variability. So, and it's kind of an interesting thing. Now, this inherent variability suggests that there are more biological possibilities than typical male and female. So if anyone ever tells you, biology tells you there's only one sex, that's just simply not true, right? There's this continuum, right? It's a spectrum. And that's not just from a biological standpoint, that's also true from a sexual from a psychological standpoint as well. So kind of this really interesting idea. And I think, as, our society progresses and becomes, more minded, this idea of sex being binary will kind of just fade to black, essentially. So now, that's sex. Sex is cool and all, but let's talk about a much more I don't know, maybe it's boring. I don't know, depends on how you view this, but let's talk about what we call linkage. Now, some genes are inherited together more often than not, and we call this phenomena linkage. So blonde hair, blue eyes, for instance, that's a gene, two traits that are more commonly inherited than other traits. The same way brown hair and brown eyes are more commonly inherited than any other combination. So this is what we call linkage. And these linked genes can become separated by recombination during meiosis. So we could sort of look at this where we have hemophilia, we have color blindness, and then we have hemophilia as well. So there's two classes of hemophilia A and hemophilia B, just as a note. But you can see that color blindness and hemophilia are carried together in this female, and then you see them pop up together here, and you see the same pattern here. Right? We have this blue-green, it pops up here, and then you have some sorting that happens, which should be an example of separation during recombination. So again, sex linkage is just things being carried next to one another. Now, this brings us to a term, and this is one of my favorite words to say. It's called synteny or syntenic genes. And synteny, or genes that are syntenic, simply means that genes that are in the same order or are located together on the same chromosome. So if two genes are syntenic, you know that they're close together on the same chromosome. Again, it's just a fun word to say, synteny. Now, for instance, we can look at red eyes and white eyes and brown bodies and yellow bodies. Now, these are syntenic genes. They're carried on the same chromosome. And you'll notice that we have this phenotype, white and brown, red and yellow, and you can have this sort of crossing, right? Now, in this case, the F1 males get only their X chromosome from their mom, and in the F1 females are those dihybrids. So this is what we're getting at here, and we can also just look at allele configurations in the F2 to the parent generation. So we originally started off with a 1-to-1-to-1 segregation in F2. I'm sorry, the deviation from a 1-to-1-to-1 segregation in F2 indicates that the genes are linked. So we have some sort of parental, we have some sort of parental generation here. There's an F1, which then produces all these F2 individuals. Now, in theory, based on what we know about genetics, they should be at a 1-to-1-to-1-to-1 ratio. But clearly they're not, right? We look at the genotypes here, it's 4,000, 4,000, 76, 53, and 9,000 individuals. So we can look at what we call the recombinant types, so red eye, brown body, and white eye, yellow body. And they have this lower abundance, so we have the lower frequency. And then we have the parental types, right? That are, this white eye, brown body, and red eye, yellow body. And there are, their percentage in terms of they're about 99% of the population. This is about 1% of the population. And you can see we have this, they're being carried together. Because again, what we would expect from this initial cross, if these were not linked together on the same chromosome, we would expect these phenotypes to be in a 1-to-1-to-1-to-1 ratio, but they're not, right? They're in basically a 48 to 48 to 1-to-1 ratio, right? So clearly not in proper links. And what is going on here is that there are, again, this idea of being linked. They're syntenic on the same chromosome, and because they're syntenic, it means they're likely to be carried together. So we can just like visualize this. It's kind of a weird concept to visualize, but let's dive in. So we have the parental generation, and they produce some offspring, males and females. So we're still thinking about sex linkage here, but what we're talking about can actually apply to shoot, spilled orange juice on my computer. Sorry, just got to clean my keyboard real quick. Let me go back. I jumped the gun by spilling orange juice. Sorry, I went really far. Let me go back. Wow, I did go really far. Sorry about that. I spilled orange juice on my keyboard. There we go. All right. Sorry about that. Definitely, after 30 years of being a human, I still don't know how to drink, apparently. So, all right, let's jump back into it. So, we have this idea. We have recessive, wild type, recessive. Well, Y chromosome here, and then there's the corresponding X chromosome there. They produce offspring, and in this case, the females are always going to be recessive wild type, and then we have wild type Y, and so on and so forth. Now, the F2 males, they're all going to be something Y, and you'll notice that the parental lines here display, essentially the same. I think the best way to think about the colors here, where we have green always stays together and light green stays together, and then we have these recombinant lines where you have a mixture of dark green and light green and dark green. Why is it recombinant? Because again, thinking about where they are, if you think about if a crossing over event happened here, it would swap this region of the chromosome for this region of the chromosome, thus producing W plus Y and W Y plus. So that's what we're getting at here. That in the absence of crossing over, we can have essentially in the absence of crossing over, two distinct alleles would be linked together inseparably, as we sort of see here in these parental types. But because of crossing over, because of these recombinant types, we have these novel phenotypes, right? This little WY and W plus Y plus, right? That gives us these novel phenotypes that are recombinant because, again, it had to arise via recombination. And if recombination didn't happen, we wouldn't see these types. Now, as we discussed, recombination happens all the time, but the probability of any given region crossing over is relatively low. So now this idea of linkage, things being tied together, can also, as I mentioned, can happen in autosomal chromosomes as well. And you can actually detect autosomal linkage by generating what we call a double heterozygote and then crossing a homozygous recessive. So it's basically a test cross, as we discussed. So in this case, we have a heterozygote BC plus and then BC plus, and then we cross with the male that's BC, BC. So B plus C, B plus C, producing a heterozygous individual. And then we do the test cross where we have this heterozygote crossing with a complete recessive individual. And we can see that we have parental classes here, recombinant classes here, again, showing that there is a favorability here, where 77% follow the parental line, and 23% are this brand new sort of recombinant line. And again, you can see that in the genetics here, or I should say the notation here. That's probably a better way to say that. And when we think about what we discussed with Mendel, that 9:3 to 3:1 ratio of a dihybrid cross is also altered because of linkage. Now, when we talked about Mendel stuff, we were looking at genes that were on different chromosomes, so there's no potential for them to be linked. So we expected that 9:3 to 3:1 ratio, but we know that's actually, when things are linked, it's not actually that 9:3 to 3:1 ratio. What we actually know is that there's just kind of this spectrum of probability where the highest frequency is up here, the lowest frequency is here, and then the lowest frequency is going to be over here. And it produces a ratio that's not 9:3 to 3:1, but we produce this greater than 9, less than 3, less than 3 to greater than 1 ratio. So we deviate from this 9:3 to 3:1 ratio because things are linked together on the sexual chromosome. Now, Franz Janssens, so in 1909, he observed that idea of the chiasmata, right, the crossing over event between chromosomes during prophase 1 of meiosis, so what we talked about last class. Now, building on Janssens' work, Morgan suggested that the chiasmata were sites of chromosome breakage and exchange, so crossing over, where chromosomes were exchanged pieces. Now, fast forward, 20 years from that, Crichton and McClintock, so they worked on corn, and Stern, who worked on Drosophila, they actually provide direct evidence that genetic recombination depends on reciprocal exchange of chromosomes. Now, if you've never heard of McClintock, Barbara McClintock, she's an awesome woman. If you want to read about a badass woman in science, Barbara McClintock is a badass woman in science. So I would recommend searching her. She's really, really cool lady. Now, they use physical markers, so the structural distinctions to identify specific markers, but we can also just use genetic markers, i.e. alleles, that we can use as points of reference for recombination. Which brings us to some kind of interesting math. It's not the most crazy math, but this does bring us to some math. So let's look at an example here. So we have parental chromosomes, females. We have a gene CAR and BAR, and there's this discontinuity here, so no genetic information. Now, meiosis occurs. Now, if there was no crossing over, we'd have CAR BAR and then the parental CAR plus and BAR plus. So the chromosomes were directly transmitted to the progeny, so nothing different, right? There's no mixing of traits here. But if things are close enough and recombination does occur, we can have that chiasmata form and a swapping of genetic information, thus producing CAR BAR plus and CAR plus BAR, right? And then this discontinuity as well. So again, novel phenotypes because these genes are close enough together, thus can be swapped via chiasmata or swapped because of crossing over. Now, the other thing about crossing over, and it's kind of an important thing, and I always forget this exists until I until I read about it, but it's kind of an interesting factor that if recombination didn't occur during meiosis 1, non-disjunction in meiosis 1 would happen way more frequently. Because of the complex that helps pair homologous chromosomes, if it wasn't for that, basically the cohesins, the proteins that hold these proteins together, couldn't separate properly, and thus non-disjunction would occur far more frequently. So it's kind of this interesting thing, right? Crossing over is important genetically speaking, but it also serves a really important role during meiosis 1, allowing chromosomes to actually physically just separate from one another, which I think is an interesting idea, right? It's essential both genetically and essential because of, you know, because of genetic diversity. So we can use this idea of how often recombination occurs, and to calculate what we call, to derive what we would call a genetic map. Now, this is produced by Sturvant. Now, he proposed that recombination frequencies, or RF, simply called, these percentages of the total progeny that were recombinant could be used as a measure of physical distance between two linkages. Now, one RF percentage is equal to 1 map unit, which is also equal to 1 centimorgan, which is just the difference to just a genetic distance. It's not like a like a centimeter. It's just a specific terminology for like describing distances between genes. It's very, very small, obviously, because chromosomes are obviously very small. So that's kind of fun. Now, the idea being is that as you increase the distances, the probability of two genes crossing over, i.e. being swapped, also decreases, right? So the closer two things are together, the more likely you are to have a crossover event. I think it's cool. So we can physically just look at this. So these are recombinant gametes. They're simply less frequent than parental gametes when they're linked. So we have this situation here. We have AB, and we have crossing over that occurs. Now, because these are so close together, when crossing over occurs at this event, they're still inherited together. AB is still here, AB is here. Now, when crossing over occurs when they're close, we have this, again, this crossing over event where we swap A for B and so on, and right, vice versa. So we produce this, again, this distinct genotype, which again leads to distinct phenotype. Now, the probability here of this occurring is more frequent than the probability of this occurring. So the recombination frequency here is low. The recombination frequency here, or RF, is high. So we can use the probability that recombination would occur to essentially get this distance, right? To draw a map of a chromosome, which I think is it's a helpful thing, right? That synteny, that order, as I suggested, is important. So, and again, we can see a recombination frequency of 50-50 here. Now we have AB again, so we have two distinct ways to write this, right? Either AA here or AA here, right? So these are very similar, just slightly swapped. Meiosis occurs when we have essentially parental gametes, so they match what we see in the parental generations, and the recombinant genes, which are, again, are a mismatch of parental alleles. Now, crossing over between the centromere and one gene, you can see that it happens here and it happens here, produces distinct distinct allele combinations, as we see here, and distinct gametes, where we have 50% is recombinant gametes and 50% is parental gametes, and we see the same thing over here. So completely independent assortment produces 50-50. Crossing over between the centromere and one gene produces, again, that 50-50 frequency. Now, the interesting thing is recombination frequencies between two genes actually can never physically exceed 50%. So the recombination frequencies of unlinked genes is 50% due to independent assortment. So if genes are not linked together, they always follow independent assortment. Thus, the frequency has to be 50%. Now, the recombination frequency of linked genes also cannot exceed 50%. So meiosis without crossovers produces only one parental chromosome, and single and double crossovers produce a 1-to-1 parental to recombination ratio on average. So that's the maximum recombination frequency we can get. And you can look at the math here if you want, but that's the key to take away. Combination frequencies don't exceed 50%. And this is kind of a handy thing, property of linked versus linked genes. And so this is a nice handy table. I stole it directly from your textbook. So that's where this is from. Now, it's kind of an interesting way that we can physically use this. And as I mentioned, we're looking at recombination frequency to map genes in physical space, right? Their orientation on a chromosome. So gene chromosome maps assign a gene to a specific location on a chromosome. This is what we call the locus. Now, the plural of this, as I'm sure many of you have heard, is loci or loci, but loci is probably the most proper way to say it. Now, we identify a gene's locus because knowing a gene's location can be used to isolate its DNA and to even understand its function. So knowing where a gene is important. And we can use many crosses to track a few genes at a time, and thus it's possible to create a gene map based upon recombination frequencies. That is the distance between genes. So now let's look at this. So we have examples here where we have multiple genes in some cases. Now, the most accurate maps are obtained by summing many small intervening distances. So left-right orientation of the map is arbitrary. So we go this way to this way or this way to this way. It doesn't matter. But we can look at gene pairs and the recombination frequency. Now, you'll notice that the recombination frequency between W and Y here is small, but the recombination frequency between W and M is significantly larger. I need to take 2 seconds. I have a puppy thing to take care of. I'll be back in 1 second. I'm so sorry.
Wait, quick question. Are we gonna be good? He's not gonna take out points because we didn't use our camera, right?
I don't know if you saw what I just commented. You don't need a camera for this class, he said in the beginning.
Oh, okay, thanks.
Sorry about that. Wife is working. Puppies have to go. Her meeting is apparently more important than my little meeting. All right. So back to recombination frequencies and mapping. So again, physical distance is related to the probability that something will be recombinant, i.e. crossing over will occur between two genes. Now remember, the probability of two genes being linked increases the closer they are. So we can physically just see that in this and D here, where W and Y are closer together. Thus, the recombination frequency is low, 1.1%. And as we increase the distance between the genes, the recombination frequency increases. And again, left to right in this map is completely arbitrary. But again, the recombination frequency, or the probability that recombination that will occur, can exceed 50%, but as you increase the distance between the genes, it increases. Because again, you can kind of think of it like if you're throwing like a ball, like through like a hoop or something, or like into a basket, right? So pretend you have a small basket. The probability of getting that ball through the small basket is low, but the probability of getting your ball into a very large basket is much higher. It's the same thing with the combination. The crossing over event to occur between two distant points is more frequent than to occur between 2 points that are very similar. So the probability of seeing this recombinant phenotype between W and Y here is lower than, say, between W and M, or even Y and M. So that's what we're going for here. We can use probability to understand how far genetic distance-wise our genes are apart. And again, this idea of linkage kind of spits in Mendel's face, right? Mendel's independent assortment says that sorting has to happen randomly, but in linkage it tells you, well, really, certain things are kind of linked together. Now, there are limitations to what we just discussed. Now, to determine gene order, if two genes are close together, it's very difficult. And actual distance between genes don't always add up. So it's kind of, you might get the wrong answer even if you do all the things right. And pairwise crosses themselves are time and labor consuming. But the best way to go about this, in this case, we were doing 2-point crosses, so between W and Y. But the best way to do this is actually with a 3-point cross. So we can do a test cross of triply heterozygous F1, so heterozygous F1 completely, heterozygous here. When we do a test cross with a complete recessive individual, we get total overall numbers. And again, we can use these recombinant types, the rarer ones, to understand their distance, because again, the closer two things are together, the more probable that they are, the less probable they are to have recombination occur. So we can physically use the results of our 3-point cross. Now, remember, these test cross progeny have 4 sets of reciprocal genotypes, and the most frequent pair has the parental configuration of alleles, so completely recessive and heterozygous here. Right, this is our parental types. Now we have recombinant types for VG relative to the parental combinations for B and PR. So we have, again, VG plus, that was our parental type, and parental type. That's kind of cool. And then we can look at recombinants for B relative to the parental types for VG and PR, and the recombinants for PR relative to parental combinations for VG and BR. So again, this is our parental types, and any deviation from that is indicative of crossing over occur, which I think is cool. Any deviation from that tells us that crossing over had to occur. And again, as probability goes down, the closer they are together. So this one is by far the least common, thus its probability is lower. Thus, we can start to actually physically calculate how far apart they are based upon these probabilities. So I took this directly from your textbook. It's figure 513. Now, we can calculate the VGB distance by simply adding up the numbers. So in this case, if you go back to this figure here, we start to add off the numbers there. So we have 252 plus 241 plus 137 plus 118 divided by the total number, which is 4,100, times 100 gives us 17%. The VGPR distance is the same idea, gives us 12%, and the BPR distance gives us 6.5%. So based upon this, we can actually remember these distances, right, are equal to our Morgan units, right, or centimorgans, right? So we can actually, just based on probability, this has the highest probability of occurring. Thus, that has to actually be the biggest distance. So this VG to B distance was most probable in terms of recombinant phenotypes. Thus, it has to have the largest distance. Because remember, the farther apart things are, the more likely recombination is to physically occur. Now, as we go down, the PR to B distance is the smallest, with PR to VG being the largest. And you'll notice that in this case, it doesn't perfectly add up, right? 12.3 plus 6.4 is 18.7, right? So that's the full distance, but we know that the distance between VG and B is 17.7. So they don't always add up, but they're not too off, right? They're only 1 mu off, right? Their recombination frequency only differs by 1%. So they're not super duper off, but they're pretty close, right? And so, but again, it's all due to probability. So if you're looking for approximate ways to get distances, it's actually a pretty good way of doing it. Now, we can infer the probable location of crossover events, again, using the same idea. So we developed our map, VGPRB. We have region 1 and region 2 here between our sister chromatids. I'm sorry, region 1 and region 2 between our genes of interest. Now, we examined the number of progeny, and we can simply compare the configuration of alleles for 2 genes at a time to the parental configuration. So let's just look at this. Now, we have a crossing over event in region 1, region 2, or between the strands here. So there's 3 potential ways for this to occur, right? It occurs between here, here, or here, or here, here, or both at the same time, I should say. Now, this is what we have. So if we have a crossing over event here, it produces this distinct genotype. Crossing over event here produces this distinct phenotype, and a double crossover produces this distinct phenotype. Right. Now, we actually have to slightly modify our calculations. Now, the initial calculation that we did was not accurate, right? Because this isn't accurate, right? 17 and 18, they're not perfectly lined up, but what our calculation doesn't make the assumption for is that a double crossover event occurs, because all we did, just to go back to this figure, we assumed that a crossover was going to happen here or here, but not simultaneously. So we actually have to go back and account for that. Now, so it gets a little complicated, but it's not a big deal. Now, this is our original cross that we did, right? 252, blah, 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 times 100 gives us 17. Now, if we correct a calculation that accounts for double crossovers, i.e. a probability of a crossover occurring, you'll notice that the math is very similar, but we add some new stuff, 13, 13, 9, 9. Where does 13, 13, 9, 9 come from? Here and here. We have to account for a crossing over event doubly occurring here. So it's where we get our 13, 13, and our 9 occurring, which gives us to 18.7, which is much more accurate. So we started with 17.7. We incorporated a double crossover event, one here and one here. We got to 18.7, which is the true distance that we observed by looking at the probability here plus the probability here. So again, when you're thinking about these crossovers, remember it can occur in one of 3 distinct ways, both or individual between your regions. And if you have to, make sure you incorporate this double crossover event to get the most accurate calculation. Now, we have this phenomenon which we call chromosomal interference, which states that occurrence of a crossover in one portion of a chromosome interferes with a crossover in an adjacent part of a chromosome. And as a note, this is not uniform between chromosomes or within a single chromosome. It's kind of random. Now, what we do is actually compare the observed and expected frequencies of a double crossover, so DCO, which is a cool little acronym, to quantify the amount of interference as what we call the coefficient of coincidence, which is actually kind of an interesting mathematical expression, and we'll get to why it's an interesting mathematical expression in a few. But the coefficient of coincidence is simply denoted as the observed DCO frequency divided by the expected DCO frequency. So the interference is 1 minus the coefficient of coincidence. So let's calculate the interference in the 3-point cross that we use. So figure 513 in your textbook, but this idea here. So the expected probability of a double crossover is the product of the single crossover frequencies in each individual. So the probability of a single crossover between VG and PR is 12.3 mu, or 0.21123. The probability of a single crossover between PR and B is 6.4%, or 0.064. Now, if the interference is equal to 0, crossovers in adjacent regions occur independently of one another. If the interference is equal to 1, no double crossovers occur. So what does this 1 mean? It just means that one doesn't affect the other. In this case, with the interference is set to 1, that there always will be interference. So double crossing over is very rare. So let's look at this. So the expected probability of a double crossover is 12% plus 6%. I'm sorry, 12% times 6%, which is giving us 0.007 or 0.79%. Now, the observed proportion of double crossovers is 13 plus 9 divided by 4,100. So remember, the 13 plus 9 is our population numbers here. Times 100 gives us 0.52%. Now, the coefficient of coincidence is this 0.52, which is our observed, divided by our expected, giving us 0.66. So the interference is 1 minus 0.66, or 0.34. So it's trending more towards this idea where the interference is likely closer to 0, thus crossover in adjacent regions occur independently of one another. But clearly there is some sort of interference here, right? You can interpret it as 34% interference, but there is some, but not a lot, right? So it's 0 to 1 scale. We're at 0.34, so we're not super tons of interference, but we're also not at completely 0 interference. So it just depends. Now, that's genetic mapping, that's interference. Now we can ask the question is, do genetic maps correlate with physical reality? Right, so we can look at these probabilities, that's one thing, but do they actually match up with what we can observe in real life? Now, we can look at physical distance in the DNA. There's actually not always a direct correspondence to genetic deformation. So we can look at the recombination frequency and the crossover frequency divided by 2, and then the physical distance between A and B, i.e. the length of DNA. The genetic distance based on the frequency of crossovers always increases. Now, but the observed RF plateaus, right? Because remember that observed RF can only be maximum of 50%. Now, there's a couple issues with this. Now, the reason these don't perfectly fall on top of one another, and they split pretty early on, is because of double, triple, and sometimes quadruple crossovers on a single chromosome. Also because 50% is the maximum, right? That's the maximum we can observe, because remember, the maximum is related to independent assortment, which is 50% maximum. And there's also what we call recombination hotspots. Sometimes some places are just more common than others, right? Recombination happens there far more frequently than others. So the idea is yes, but also no. So, but they're still pretty good, right? They're still pretty good. Now, Drosophila melanogaster has what we call 4 linkage groups. Now, genes that are connected by linkage relationships are what we call linkage groups. So when there are many genes per chromosomes, when many genes per chromosomes have been mapped by a linkage, the group is synonymous with a chromosome. So, so we can look at chromosome, different chromosomes here. Drosophila only has 4 chromosomes, just as a note, but we have the X chromosome, chromosome 1, chromosome 2, and then other chromosome, forget its name. Now we can look at chromosome X, and you can have all these genetic distances that, again, are related to recombination frequencies, and different traits are linked based upon where they are on these chromosomes. So these are what we call linkage groups because they're found together. They've been mapped, so they're synonymous with this chromosome. That's all. So now this brings us to an important mathematical concept, which I think is probably the most complex mathematical concept we'll get into for this course, which is what we call a chi-square test. And it's a relatively important mathematical test for genetics, and as a general rule. Now, what we can do is we can define an expected ratio, and in our case, the expected ratios, typically speaking, is 1 to 1 to 1. And any deviation from that expected ratio of 1 to 1 to 1 to 1 can represent chance events or linkage. Now, this chi-square test measures what we call the goodness of fit between observed and expected values, which leads us to a concept that you've probably heard of, which is the null hypothesis, which is the observed values are no different than expected values, right? That our observed values are going to be 1 to 1 to 1 to 1, and our expected values are also 1 to 1 to 1 to 1. Yeah. Now, when we're thinking about genetic linkage, in linkage studies, the null hypothesis is that there is no linkage between two genes. And if the genes are not linked, we expect that 1 to 1 to 1 ratio in the F2 progeny, as Mendel showed us, right? However, we can use a chi-square test to reject this null hypothesis, but it actually can't provide us with a hypothesis. So that's kind of an important thing, right? When you think about what hypothesis testing is commonly used for, it's usually to reject your null hypothesis and accept your hypothesis, or to reject your hypothesis and accept the null hypothesis. In the case of a chi-square test, in terms of genetics, we can really only reject our null hypothesis. And our null hypothesis is that there are no differences from expected values. So there's no deviation from Mendel's, independent assortment. So that's how we use a chi-square test. So we can use a chi-square test, and you'll do this in your lab, actually you do it on a couple different things, but you use this in your lab. So we use our data from an experiment, and we need a couple of information. So we need the total number of progeny, how many classes of progeny, and the total number of offspring observed in each class. And what we could do is calculate the number of offspring expected in each class if there is simply no linkage. So the chi-square formula, and you might have seen this before, chi-square, so this is chi, the weird kind of x, is equal to the sum, that's what the Greek letter E stands for, the number of observed minus the number expected. So if I expect 1, if I observed 1 and my expected is 1, that's what I'm getting at. We square that, divided by the number expected, or simply written as the sum of O minus E squared divided by E. So that's the chi-squared test, the sum of O minus E squared divided by E. Now, we have to consider what we call the degrees of freedom, because all we get out the back end of this equation, as you can see, is the chi-squared value. Now, the chi-square value is important, but it doesn't really tell you very much without understanding degrees of freedom. Now, when you took statistics, you probably learned what a degree of freedom is, but I'll just reiterate exactly what it means. When we say degree of freedom, it's the mathematical consideration for the number of independent varying parameters. So you define degree of freedom is n minus 1, where n is the number of classes. Now, what do I mean by classes? Classes of progeny, so different phenotypes, different genotypes. That's what we're getting at. And from this, we pair the chi-square value with the degrees of freedom to determine a p-value or a probability value. And what it's really telling us is what is the probability that the deviation from expected results had occurred by chance. So basically speaking, when we think about what we get out the back end of this, is a probability that we can accept or reject our null hypothesis. So if that probability is less than 0.05, we have to essentially reject our null hypothesis. if that probability is greater than 0.05, we have to accept that null hypothesis. So that's kind of this threshold what we use, this 0.05, a 1 in 20 or a 0.05 chance of us being able to accept or reject our hypothesis. In this case, for chi-squared, in genetics, it is simply that null hypothesis. So let's apply the chi-square test to see if genes A and B are linked. So we have the parentals and we have the recombinants, right? Big A, little b, big B, right? That's the parentals are A, B, and little a, little b. So pretty straightforward. Now we look at the observed, which is 18, 14, 7, and 9 for a total of 48 offspring. Now we expect, based on Mendel, a 1 to 1 to 1 to 1 ratio. So if we have 48 individuals, our total has to still be 48, but it has to be, each one has to be in a 1 to 1 to 1 ratio, which has to be 12 to 12 to 12 to 12. Now, that's fun. Now we do the math. So observed, right? 36, observed minus expected. So 18 minus 12 squared gives us 36 divided by 12. And then we do the same thing for all of those. Now remember, we have to sum these all together. So we sum all of these together, and that gives us our chi-square value. In this case, that chi-square value is 6.17. Now, we get a degree of freedom by looking at all the potential genotypes in this case. There's 1, 2, 3, 4. 4 distinct genotypes. Now remember, we know that degree of freedom says the number of classes minus 1. So we know we have 4 distinct genotypes. We minus 1 gives us 3 degrees of freedom. So in this case, we have a chi-square value of 6.17, 3 degrees of freedom, and our p-value, which we would, here we go, which we would determine from a chi-square table, and I'll show you this chi-square table in a second, but this is what we would look for. We use it to determine our p-values. Oops, wrong way. And we can see a second experiment where we have observed is 36, 28, 14, and 18. Total is 96, but again, our expected is a 1 to 1 to 1 ratio. So we have to put 24 in because 24 times 4 is 96. In this case, we had a chi-square value of 12.3 and a p-value of less than 0.01. Now you'll notice there's a relationship. The larger the chi value is, typically the more likely you are to get a significant value. And why is that? Well, let's look at what we call a chi-square table. Now this is a very simplified chi-square table. They get much more bigger and complex, but remember, we use the p-value of 0.05 as a cutoff. And so the way to interpret is that there's less than 0.5% or 0.05 or 1 in 20 chance of attaining the same results by random sampling if the null hypothesis is true. So the lower our p-value, the more likely, the more confident we can be in our answer, essentially. Now, again, and it also ties back to our capacity to reject the null hypothesis. We use this critical threshold of 5%, that if it's greater than that, we have to accept the null hypothesis. Right? We can't reject it. If it's less than 0.05, we have to reject the null hypothesis. Now, there's a lot more that goes into this, and that would be better served for a statistics course, which you all have presumably taken at this point. So, but that's how we're interpreting p-values in terms of a chi-square test. What is the probability based on our capacity to accept or reject the null hypothesis? Now, you'll notice how this chi-square table is set up. We have p-values here at the top. The white here is can't reject the null hypothesis. Yellow is we can reject the null hypothesis. Then we have degrees of freedom on this column here, and each degree of freedom corresponds to a single row. And what's in here is chi-square values. So if we go back to our thing, we got 6.17. So we go, okay, how many degrees of freedom do I have? 1, 2, 3. And then I find 6.17. 6.17 isn't on here, but it's between 6.25 and 2.37. Thus, my p-value has to be greater than 0.1. We go back to our other one. The chi-square value is 12.3. We had 1, 2, 3 degrees of freedom. We try to find 12.3. It's not on here, but it's between 11.25 and 16.27. Thus, our p-value has to be less than 0.01. So the idea, remember, with chi-square, we're mathematically testing is if what we observed is due to random chance or not. So can we reject the null hypothesis? Can we reinforce that there is linkage here? Because in this example, there's linkage mathematically supported. In this example, it's not mathematically supported that there is linkage. That's what we're getting at here. And you can use a chi-square test to test a number of different things. It doesn't have to just be linkage. You can test if it deviates from a 9:3 to 3:1 ratio. You can test anything with a chi-square test, as long as you have an expected value and you observe some sort of variables, which is nice about chi-square tests. You can, like I said, you can use it for a number of different things, right? If you expect this and you get this, is there a mathematical difference between the two? Which is nice. So that's linkage. Now we have this kind of last bit of today's class where we're looking at a couple of funky things in eukaryotic biology. And so the first one's going to be what we call tetrad analysis in our fungi. Now, as many of you probably know, fungi exist primarily as haploid and multicellular organisms. And so they're kind of unique, right? Because, well, mostly when we think about animals, right, we exist as diploid organisms, but these organisms exist as haploid and multicellular. So we're going to talk about two really interesting species of fungi today, one of which you probably know and love, which is Saccharomyces cerevisiae. You'll recognize Saccharomyces as a model organism, but it's also, what makes beer and bread and all the tasty fermented things out there. Now we also have Neurospora crassa, which is a mold that grows on bread, so he's not your friend, if you like bread, of course. So two distinct yeasts, Saccharomyces being single cellular, Neurospora being multicellular. And it's kind of interesting that after meiosis occurs, all four haploid products of each organism's meiosis are contained with what we call an ascus, or just a sac. You can call it either way you want. And this produces what we call ascospores or haplospores, so haploid spores. And it's interesting that these can germinate and survive as viable haploids that then divide by mitosis, or where you have tetrads that are four ascospores in a single ascus or sac. It's just kind of weird. But let's look at Saccharomyces cerevisiae. So we have the ascus containing four haploid ascospores. Now there's A mating types where an ascospore is released, and that ascospore germinates, goes into a vegetative haploid life cycle where it buds into, as its division. Or it can go into an alpha mating type where the ascospore is released, it germinates into a vegetative cycle that's also haploid. So two distinct types. You can think of them like, sperm and egg almost. Now what's cool is if you have an alpha meet an A, they can form a zygote. They can fuse together, form a zygote with one another. The zygote forms, it buds, and then that bud, that A alpha mixture, then goes on to perform meiosis, thus producing the ascus, thus repeating the cycle over and over again. So it's kind of this weird system, right? Where your adult is haploid, and then you have this kind of odd cycle where they fuse together, producing a diploid. Then they produce this ascus that has four haploid cells in there, which is kind of weird, right? It'd be the functional equivalent if you produce eggs or sperm and they just live together in this big thing and that's all you were, and then you release them from time and time again to reproduce. It's kind of this weird system. But that's Saccharomyces. Now we can also look at what we call there's three distinct tetrad types in a dihybrid yeast. Now haploid strains of opposite mating type, so A and alpha, can be mated and the resulting diploid to undergo meiosis. So if we have haploid parents, big A, big B, and little a, little b, so A type and alpha mating type, the diploid cell is heterozygote, which undergoes words right on there, meiosis, to produce what we call the non-parental ditype, little a, big B, little a, big B, big A, little b. Now we can also produce unordered tetrads, which We have the parental type, which is AB and AB, or the tetratype, which has all four. Right? It has a mixture of parental and haploid types, which is kind of interesting. And that again, that's what we see in our weird kind of fungi. So odd sort of scenario, not typical of what we see in the genetic world, but it is present and something that we should definitely be aware of as a concept. As a general note, as you move into the world of fungi, things get weirder and weirder and weirder, which is why we don't talk about fungi a lot in this course. But they do get really weird. Fungi do a lot of weird stuff genetically that other organisms simply just don't do. Now, the phenotype of a haploid fungi is directly representative of their phenotype. That makes sense. If you only have half your number of chromosomes, you're always going to express your genotype, right? That's how it works. Now, mutations in haploids can also affect the appearance of cells and their capacity to grow under certain examples. So a couple different mutants that we can look at. So HIS4, mutant's recessive, and it's actually unable to grow in the presence of histidine. HIS4, which is the dominant allele, grows in the presence or absence of histidine. And the same thing for TRP. So we have a TRP1 mutant, which is recessive, and it's unable to grow in the absence of tryptophan. And then TRP1, which is dominant, can grow in the presence or absence of tryptophan. So it's kind of an interesting system where you just need one physical copy of the gene, because you're haploid, to display the trait. So it makes things a little bit easier. Now, we can generate diploid yeast for our genes, right? Because we can fuse an alpha and an A together, and that to thus produce a diploid organism. So we could do a cross here. We have HIS4 TRP1 and HIS4 TRP1 A and alpha, thus producing HIS4 and HIS4, and then TRP1 and TRP1 A alpha. So we have this diploid organism that is again a hybrid, right? A dihybrid, right? It's got the recessive and the dominant together, which is pretty cool. Now, meiosis itself can generate 3 distinct kinds of tetrads from that. As we saw in a previous slide. Now, a parental ditype, or PD, all spores with the parental allele, which is 0 out of 4 recombinants, which we see here. But our tetrads can be unique, right? HIS4 TRP1, HIS4 TRP1, and then we have that combination, right? So we have 2 parentals, 2 recombinant lines here. So, and then ultimately the ditype that we produce from this is going to be a combination. And then so this is our parental ditype. We can produce non-parental ditypes, as we saw a few slides ago. And then finally we could produce the tetratype, where 2 have parental alleles, 2 have recombinant alleles, and there's a crossover between the centromere and the closest gene. So unique fungal genetic structures can produce some novel tetratypes here. Again, similar to the same idea, right? Where we can have crossing over, bringing together novel genotypes together, and again, all dependent on how close they are on the chromosome together. Now, when we can actually apply this idea of linkage to our friend fungi here. Now, when genes are unlinked, the number of PD essentially equals the number of NPD. So remember, non-parental ditype and parental ditype. So PD, so the number of tetrads we produce, 31, 28, and then the tetraploid is 41. So when the genes are unlinked, they should be equal to one another, and they're about equal to one another. As you can see, 31 is pretty dang close to 28. Now, when genes are linked, the number of PD greatly exceeds the number of NPD. So we can look at a different example here. We have ARG and URA, the A mating type and the alpha mating type. They produce a diploid cell, and we can look at the products of meiosis, where we have 127 PD, 3 NPD, and then 70T. And you can see the number of PD greatly exceeds the number of NPD, right? So, which tells us that they're linked with one another, that the probability of swapping ARG, capital for ARG lowercase, is extremely low. So the physical distance on these, remember, in terms of recombination frequencies, would tell us they're very close together. So similar to what we saw. And you can physically see an example of what this looks like, right? So no crossing over, single crossing over, where you have, again, that exchange of genetic information here, producing these novel genotypes, which is leading to either a tetratype in this case, or the parental type where there is no crossing over. Right? So again, tying back to this idea of genes being linkage, crossing over being a capacity to exchange genetic information between A and B. We can also look at double crossing over, or DCO, where there's 2 double crossing overs, or 3-strand, which is rare but still occurs. But again, we're producing, if with a double crossover event, we're producing the parental ditype, because again, we had a double crossover, but nothing meaningful was exchanged in terms of swapping genotypes. And then we can look at a 3-strand double crossing over event, which produces one of our tetratypes. And same thing, 3-strand double crossover event can produce a tetratype, and a 4-strand double crossover event can produce a non-parental ditype. So again, like in normal eukaryotes, our fungi, which have this weird genetic system, can again produce, can have genes that are linked together. And very much like our normal eukaryotes, we can calculate recombination frequencies, which we simply do it as NPD plus 1/2 of the number of tetrads divided by the total number of tetrads times 100. So for figure 522, which is this figure, but it's also figure 522 in your textbook, recombination frequency is 3 plus 1/2 times 70 divided by 200 gives us 19 mu. So again, similar idea that we can use crossing over to calculate, recombination to calculate how far apart physical distance things are from one another. Right. And I'm actually going to end things here. I don't think what I have left here is all that important, but I wanted to highlight what we went through in terms of how you can apply this system to a non-conventional eukaryotic model system that we see with yeast, to understand how linkage and how recombination themselves are physically related. So if you want to take a peek at some of this other stuff, looking at neurospora, same stuff that we just talked about, you can take a peek at it, but I will let you do that on your own, because again, I just wanted to highlight that this idea of linkage and calculating distances is actually can be applied to a sort of a non-traditional system as well. So with that, I'm going to stop recording. If you guys have any